Hey everybody, so today I'm going to be talking about intracellular calcium signaling pathways. Now this video is going to be quite advanced, so if you are unfamiliar with a lot of the terms that I use, I recommend that you go back and watch some of my previous videos. Now, the first synapse that we're going to be talking about is the SP5, or the spinal trigeminal nucleus, synapse onto the fusiform cells in the dorsal cochlear nucleus. And today we're going to explore how, for example, excitatory postsynaptic potentials, or EPSPs, and NMDA receptor activation at the SP5 fusiform synapse can lead to intracellular calcium signaling cascades. Regulating, of course, the AMPAR receptor trafficking for long-term potentiation or long-term depression. Now let's talk a little bit about the EPSPs and NMDA receptor activation at the SP5 fusiform synapse. Now, if we're talking about glutamatergic EPSPs, the activation of SP5 efferents releases glutamate onto fusiform cell dendrites, generating an EPSP mainly through AMPA-type glutamate receptors. And the AMPA receptors rapidly depolarize the postsynaptic membrane by gating sodium or potassium. And it produces a fast EPSP. And if the EPSP is strong or repetitive, it can depolarize the fusiform cell sufficiently to engage NMDA receptors. Now, the NMDA receptors at this specific synapse are ionotropic glutamate receptors that require the dual activation by ligand binding and postsynaptic depolarization. Now, as we all know, at resting potential, NMDA receptors are blocked by magnesium ions in a voltage-dependent manner. And when AMPA receptor-mediated depolarization, either from the ANF, the auditory nerve fiber, or from the SP5, pushes the membrane potential upward, the magnesium block is relieved, allowing NMDA receptors to conduct. Basically, the NMDA receptors act as coincidence detectors, and they open only when presynaptic glutamate release coincides with sufficient postsynaptic depolarization from either sides. As we know, fusiform cells have both apical dendrites and basal dendrites. Apical dendrites are the dendrites on pyramid neurons, like the fusiform cells, that extend or project upwards from the soma, or they extend from the apex of the cell body, and project, of course, like I mentioned, upwards. And the basal dendrites are the opposite, or the ones that project to the auditory nerve fibers. And an EPSP that arrives from the auditory nerve fiber can be sufficient enough to, depolarize the fusiform cell to such an extent that the magnesium block is removed from the NMDA receptors, allowing for the low sustained EPSPs from the SP5 pathway to promote the low sustained calcium spiking required for long-term depression. Now, as I mentioned before, the NMDA receptors act as coincidence detectors, and they open, of course, only when these thresholds are reached, or when these requirements are fulfilled. Yes, the presynaptic glutamate release coinciding with, of course, the postsynaptic depolarization. Now, this dual gating mechanism ensures that the NMDA receptors and the calcium influx that they carry occur primarily during strong or coincident activity. Now, once the magnesium block is lifted, of course, the NMDA receptor channels admit the calcium along with some sodium into the fusiform cell, and this calcium entry is a very important trigger for downstream signaling. And NMDA receptors, of course, also require the binding of two coagonists, specifically glutamate and glycine, or D-serine, to open. But under physiological conditions, glycine or D-serine is usually available extracellularly. So a large EPSP or multiple EPSPs in close succession is typically needed to recruit the NMDA receptors. Now, this interplay of AMPA and NMDA receptors makes the EPSP biphasic, so to speak. So an early fast depolarization via the AMPA receptors, followed by a slower NMDA component if the depolarization was sufficient. And this arrangement or organization is super duper important at the SP5 fusiform synapse for gating plasticity. That means that pretty much only combined somatosensory presynaptic activity and postsynaptic firing will elicit some very substantial NMDA receptor activation. And in the case of tinnitus, there is some aberrant somatosensory signals constantly from the SP5 pathway, and rapid firing of the fusiform cells in general would be, really rapid firing from, for example, high frequency noise or just loud noise in general. So very rapid and very strong EPSPs would be sufficient to induce long-term potentiation just due to this presence of very strong signals, nerve signals, so to speak. Now, as we know, if we're talking about spike timing dependent plasticity in the DCN fusiform cells, the synapse follows the typical Hebbian spike timing rules governed by the NMDA receptors. So if a somatosensor EPSP or the SP5 input precedes the fusiform cell spike, the NMDA receptors open and trigger long-term potentiation of the synapse or the Hebbian pairing. Now, on the other hand, if the fusiform cell fires before the somatosensory EPSP arrives, or the reverse order or anti-Hebbian timing rules, the NMDA receptor activation is minimized and long-term depression is induced. And this spike timing dependent plasticity is partially NMDA dependent. So blocking the NMDA receptors would disrupt the timing rules and, of course, the resulting plastic changes as well. Now, interestingly enough, the reason why some people have exceptionally stable tinnitus would be potentially due to a lesser prevalence of somatosensory cofactors, for example. If you remember what I said previously, generally LTP is dependent on the presynaptic activity occurring before the postsynaptic activity, meaning the SP5 EPSP occurring before the fusiform cell spike, for example. And this, of course, is necessary for the induction of long-term synaptic potentiation. So if there is minimal or almost no presynaptic EPSPs from the SP5 pathway, that means that achieving even LTP would be substantially more difficult. Not impossible, but difficult. And this is, of course, why other cofactors like stress, which promotes the release of cortisol that sensitizes the neurons, and inflammation, which also sensitizes the neurons, pro-inflammatory cytokines, for example, and messing with 5-HT pathways or serotonin pathways also leads to more excitability in specifically at this synapse. And this is why all of these cofactors can potentially make tinnitus easier to develop or easier to worsen due to the EPSPs, the presynaptic EPSPs being more prevalent. Basically, NMDA receptor-mediated calcium entry at the SP5 fusiform synapse is a prerequisite for synaptic strengthening and synaptic weakening. Now let's talk about how calcium influx is translated into molecular signals. Now, some of you are confused as to why there is a low sustained calcium influx during LTD if the fusiform cells spike first. That is due to the repolarization or the hyperpolarization or refractory period of the fusiform cell, meaning that after the fusiform cell spikes or the action potential spikes, it starts going down and the membrane potential becomes way more negative than the resting membrane potential. And in this specific time frame, or during this specific time frame, the membrane potential or the EPSPs cannot initiate a strong, response from the fusiform cell. So just because the fusiform cell's membrane potential is more negative than the resting membrane potential, this means that the fusiform itself, the fusiform cell itself, is very, very unlikely to spike from the EPSPs, from the SP5 synapse, and the calcium influx in general would be very low because of this. Now, let's talk about intracellular calcium signaling cascades. Now, calcium, or the calcium ions as a second messenger, enter through the NMDA receptor channels and through voltage-gated calcium channels. If the cell spikes, it initiates the complex intracellular signaling. Now, it binds to calmodulin, or CAM, which is a ubiquitous calcium ion sensor. And it changes the CAM's conformation, allowing it to regulate downstream enzymes. Now, two of the most important calcium or calmodulin targets in synaptic plasticity are calmodulin-dependent protein kinase 2, or CaMKII, you guys might recognize it, and the phosphatase calcineurin, or CAN, also known as PP2B. Now, the outcome, LTP or LTD, is essentially determined by the balance of kinase versus phosphatase activation, which in turn depends on the, of course, spatiotemporal profile of the calcium ion signal. Now, high-frequency stimulation, or tightly timed pre-post spikes, produce a large rapid calcium rise that strongly activates CaMKII and related kinases, biasing towards LTP. And on the other hand, low-frequency stimulation yields a slower, lower amplitude calcium elevation that preferentially activates calcineurin and other phosphatases, favoring LTD. And this calcium-based threshold model is, of course, supported by both experimental and computational studies. Now let's talk a little bit about the kinase pathways for LTP induction. A large calcium transient will rapidly saturate calmodulin and activate CaMKII, which then autophosphorylates at THR286 to become autonomously active. And the activated CaMKII translocates to the postsynaptic density, or PSD, and phosphorylates target proteins that enhance synaptic strength. We're going to talk more about this in the next section. And calcium can also stimulate protein kinase C, or PKC, in conjunction with G protein pathways. For example, group 1 metabotropic glutamate receptors, or mGlur1/5, often co-localized at fusiform cell synapses, activate GQ/PLC when glutamate binds. And the PLC produces inositol trisphosphate, which you guys might recognize as IP3, and diacylglycerol, or DAG, inside the cell. IP3 releases calcium ions from intracellular stores, or the endoplasmic reticulum, by binding IP3 receptors, while DAG, together with calcium ions, co-activate conventional PKC isoforms. And the rise in cytosolic calcium can be amplified by internal calcium release. And PKC becomes another calcium-sensitive kinase contributing to plasticity. And in parallel, calcium, or CAM, may activate adenylyl cyclase, or the AC1 isoform, increasing cAMP and protein kinase A, or PKA, activity. And PKA can phosphorylate AMPAR receptors, in other words, at GLU-A1 S845, and other proteins to promote LTP maintenance. So if you guys didn't really understand what I just said, basically LTP is self-reinforcing and self-sustaining, not only from external factors, but from also intracellular mechanisms. And basically the high calcium influx engages this massive network of kinases, the CaMKII, PKC, PKA, that all converge on strengthening the synapse. Now let's talk about the sexy phosphatase pathways for LTD induction, exactly what we want with the Susan Shore device. When calcium levels are modest or rise slowly, calcineurin, or CAN, is preferentially activated. Now, calcineurin is, of course, also a calcium or CAM dependent serine, or threonine phosphatase, that upon activation dephosphorylates multiple substrates, including the key synaptic proteins. One direct target is, of course, the AMPAR receptor subunit GLU-A1, which we mentioned before, SER845. Calcineurin removes this PKA-added phosphate, which is, of course, associated with AMPAR receptor removal from the synapse. Calcineurin also dephosphorylates inhibitor 1, or I1, or DRPP3-2, relieving their inhibition of protein phosphatase 1, or PP1. And freed from the inhibition, PP1 can further dephosphorylate AMPAR receptors and postsynaptic scaffolding proteins, contributing to LTD. The coordinated action of CAN and PP1 broadly dephosphorylates the postsynaptic targets, basically counteracting the kinase effects. Importantly, PP1 can even dephosphorylate CaMKII at THR286, which terminates the CaMKII's autonomous activity and the overall LTP processes. And in fusiform cells, this kind of push-pull or tug-of-war between CaMKII and CANPP1 determines the direction of plasticity. If you guys, of course, remember, the CaMKII is LTP and calcineurin is LTD. And the cell basically reads the calcium signal duration and amplitude to engage the appropriate enzymic pathways. Now, there's also metabotropic modulation that people don't really think about too much. The metabotropic glutamate receptors, or the mGlurs, and other G-protein coupled receptors, they fine-tune the calcium signaling. Now, at the sp5 to fusiform synapse, the spillover glutamate, or high-frequency stimulation, can activate the mGlur1/5, which, as I mentioned before, releases the internal calcium and activates the PKC. Now, this, of course, can happen for people even without significant somatosensory issues or somatosensory irritation. For example, somebody with non-somatic tinnitus, which means that, their somatosensory cofactors are not so strong, can still spike from the activation of mGlur1/5 or the internal calcium stores. Yeah. Now, this can technically also facilitate LTD induction, especially in conjunction with the moderate NMDA calcium influx, which is actually very similar to cerebellar LTD, which we will talk about later if we have time. And the group 2 or group 3 mGlurs on pre or postsynaptic, on the pre or postsynapse, they inhibit adenyl cyclase and reduce the CAMKII, which might dampen the LTP maintenance, actually. So neuromodulators like acetylcholine or norepinephrine, which act on G_Q or G_S coupled receptors, can also modulate calcium dynamics and kinase or phosphatase balance. So while NMDA receptors provide the primary calcium trigger, these second messengers like IP3, DAG, or CAMP, and metabotropic pathways, they shape the intracellular calcium landscape, and of course the resulting plasticity outcome. Now let's talk about AMPA receptor trafficking in LTP and LTD. Now, first of all, about the basal AMPA receptor composition. Now, AMPA receptors at the central synapses are tetramers, which usually contain GluA1/4 subunits. Fusiform cells, like most neurons, predominantly express GluA2 containing AMPA receptors at rest. And the GluA2 subunit renders the receptor impermeable to calcium due to the RNA-edited arginine of the QR site. So under base conditions or under basal conditions, EPSCs at the SP5-2 fusiform cell synapse are mediated by calcium-impermeable AMPA receptors and do not themselves raise postsynaptic calcium. That job is left to the NMDA receptors. However, the subunit composition of AMPA receptors can change during plasticity. And neurons, they maintain pools of AMPA receptors in the endosomes that can be delivered to or removed from the synapses. And the trafficking of these receptors to and from the postsynaptic membrane is the primary mechanism, of course, for the expression of LTD and LTP. And in LTP, more AMPA receptors are added to the synapse, increasing synaptic strength, while in LTD, the AMPA receptors are internalized, weakening the synapse, and hopefully dissolved later on. And of course, the many proteins regulate this trafficking, including the receptor subunits phosphorylation, auxiliary proteins, and the endocytic machinery. Now, if we talk about LTP, or the AMPA receptor insertion, during this process, a burst of calcium, or CAM kinase activity, leads to the phosphorylation of the AMPA receptor subunits and, of course, their associated proteins. For example, CaMKII phosphorylates GluA1 at SER831, which increases the channel conductance of AMPA and promoting synaptic insertion, and pKa phosphorylates the GluA1 at SER845, which primes the receptors for recruitment to the membrane. And in parallel, CaMK2 phosphorylates stargazin, which is a TARP, or TARP, or transmembrane AMPA regulatory protein, also known as gamma-2, which is an AMPA receptor auxiliary subunit in the postsynaptic density. Phosphorylated stargazin has reduced interaction with membrane lipids and increased binding to PSD95, which effectively traps the AMPA receptors in the synapse. And this CAMK2-dependent stargazin phosphorylation greatly reduces AMPA receptor lateral diffusion, which stabilizes more receptors at the synaptic site. And so basically the net effect is that the AMPA receptors from the reserve pools are delivered and locked into the postsynaptic membrane. And some of these inserted receptors during early LTP are GluA1 homomers, lacking the GluA2. In other words, the calcium permeable AMPAR receptors, or CP AMPARs, we'll call them. Now, the CP AMPARs can carry additional calcium into the spine on subsequent synaptic events, which may further activate kinases and stabilize the potentiation. And within minutes, most inserted AMPAR receptors at mature synapses become GluA1/A2 heteromers. Now, the AMPAR receptor insertion in LTP relies on vesicle trafficking. After induction, the receptors are exocytosed from recycling endosomes to the extrasynaptic membrane. SNARE, or SNARE, proteins as we'll call them, like syntaxin 4, help fuse these vesicles at specific membrane domains. And the receptors then diffuse laterally into the PSD and are tethered by scaffolding proteins like PSD95, aka P79, or GRIP, or GRIP. Now, importantly, an ATPase called NSF, or anaethylamide-sensitive factor, binds to GluA2 subunits and assists in releasing AMPAR receptors from certain interactions to permit their mobilization. Blocking NSF activity has been shown to prevent LTP expression, which is important, which indicates that NSF-mediated trafficking is required to finalize the AMPAR receptor incorporation. And of course, through these steps, all these steps, the synapse gains the additional AMPAR receptors, making the subsequent EPSPs larger, or the potentiation. And consistent with this, fusiform cell LTP is expressed as an increase in AMPA-EPSC amplitude and involves the NMDA-dependent recruitment of AMPAR receptors. For those of you who are confused about what EPSC is, EPSC refers to the excitatory postsynaptic current, and AMPA-EPSC is the excitatory postsynaptic current mediated by AMPA receptors. Now let's talk about LTD, or the AMPA receptor removal. Now, of course, the opposite process occurs to LTD, the opposite process to what I described above. The moderate prolonged calcium levels activate the phosphatases and often the protein kinase C via the mGlur-mediated DAG. That act on the AMPA receptor complex proteins to trigger endocytosis. A really important event in many forms of LTD, including at the parallel fiber synapses, is the phosphorylation of the GluA2 subunit at SER880 by PKC. Now, this site is in the PDZ binding tail of GluA2, and when phosphorylated, it reduces GluA2's affinity for GRIP, or GRIP. Which, if you guys remember, is a synaptic anchoring protein, and it increases its affinity for PICK1. Now, PICK1 is an AMPA binding protein that contains a BAR, or BAR domain, which helps induce membrane curvature for endocytosis. And by switching the binding partner from GRIP, which retains AMPA receptors at the synapse, to PICK1, SER880 phosphorylation effectively earmarks AMPA receptors for removal. And concurrently, calcium-activated phosphatases can dephosphorylate GluA2 at TYR876, or TYR876, which permits an interaction with the endocytic adapter BRAG2, or BRAG2, a guanine nucleotide exchange factor for ARF6 GTPase. Now, activated ARF6 then assists the clathrin-mediated endocytosis of AMPAR receptors. And AMPAR receptors are removed from the synapse via clathrin-coated pits adjacent to the PSD, with AP2 adapter complexes recognizing the receptor tails, especially when modified by phosphorylation or ubiquitin. Now, once internalized, the AMPAR receptors enter endosomal compartments. Now, here, another layer of regulation occurs. Ubiquitination of AMPAR receptor subunits directs their post-endocytic fate. Now, brief neuronal activity can rapidly add ubiquitin molecules to AMPAR receptor subunits, GluA1 and GluA2, on specific lysine residues. In other words, LYS868 on GluA1 and LYS870/882 on GluA2. And this activity-dependent ubiquitation is calcium-dependent and requires CaMKII activity, surprisingly. And while ubiquitination does not appear to affect the initial endocytosis, the internalization occurs normally, even if these lysins are mutated, for example. It controls whether internalized receptors are sent for degradation or recycled. Now, this is really, really important. So, ubiquitinated AMPAR receptors are more likely to be sorted into late endosomes or lysosomes for proteolysis, thereby reducing the total receptor pool. Now, in LTD, persistent low-level stimulation can engage this pathway to permanently reduce AMPAR receptor numbers, or for them to be dissolved, which is exactly what we need. Now, if the stimulation is mild or transient, internalized AMPAR receptors might avoid ubiquitination and be recycled back to the surface later, resulting only in transient depression. In fusiform cells, prolonged somatosensory input paired with auditory, for example, deprivation, leads to sustained AMPAR receptor removal and degradation, consistent with enhanced LTD and reduced synaptic gain. But this is only exclusively in animals, in the study that Susan Shore did, that I quoted, animals resilient to tinnitus. So the somatosensory input induces long-term depression in the DCN and prevents the pathological plasticity. I assume this is due to the scrambling of the synchrony. But prolonged somatosensory stimulation, or somatosensory-only stimulation, produces long-term potentiation in the tinnitus-specific models. And this is, of course, due to the messed up excitatory-inhibitory balance in the tinnitus model. Now, if you take a look at the diagram, you can see that the AMPAR receptor trafficking during LTD is on the left in green, and LTP is on the right in yellow, at the SP5 fusiform cell synapse. Now, during LTD, as you can see, the prolonged low-level calcium influx, or the small red dots, activates the phosphatase and PKC, which phosphorylates the GluA2 at SER880. And this causes the GluA2 to detach from the anchoring proteins, the GRIP or GRIP, and bind to the PICK1, promoting the AMPA receptor clustering in presynaptic endocytic zones. The AMPA receptors, which are in red, are then removed by the clathrin-mediated endocytosis. Now, the dephosphorylation of GluA2-TYR876 and activation of the ARF6 GTPase via the BRAG2 also facilitate the internalization. And internalized receptors may be tagged with ubiquitin, or the small gray tags, for degradation in lysosomes. And during the LTP process, the high frequency activates a large calcium influx, or the yellow sparkles, as you can see, through the NMDA receptors, the GLUN, which are teal colored, and newly inserted calcium-permeable AMPAR receptors. And this strong calcium activates CaMKII, PKC, and pKa, which phosphorylate AMPAR receptor subunits, in other words, GluA1 at SER831 by CaMKII, SER845 by pKa. And phosphorylation, which is shown as the blue P tags, triggers the exocytosis of AMPAR receptors, which are green on that part of the diagram, from recycling endosomes into the membrane. NSF and SNAPs, or SNAPs, assist in this fusion. And the AMPAR receptors then move laterally into the PSD and are stabilized by binding to scaffolds, the PSD95, etc., and TARPs, or TARPs. CaMKII also phosphorylates the TARP, or starkazin, enhancing AMPAR receptor retention at the synapse. And the result is an increased density of AMPAR receptors at the synapse, strengthening the excitatory postsynaptic potential. Now, if we talk about the key molecular players, several specific proteins orchestrate the AMPAR receptor trafficking. One being TARPs, in other words, starkazin / gamma-2, auxiliary subunits that link the AMPAR receptors to PSD scaffolds. The phosphorylation of TARPs by CaMKII during LTP increases AMPAR receptor anchoring. The PICK1 binds to GluA2 when SER880 is phosphorylated, critical for AMPAR receptor endocytosis in LTD. And in some experiments, mice that lacked the PICK1 have impaired LTD, as AMPAR receptors cannot be readily removed. And the GRIP1/2, the PDZ domain proteins that hold the GluA2 containing AMPAR receptors at the synapse. Now, during LTP, high GRIP, or GRIP, binding helps maintain the newly inserted receptors. And during LTD, GRIP, or GRIP, is displaced by PICK1. And NSF, an ATPase that uncouples GluA2 from PICK1/GRIP complexes, allows the recycling or reinsertion of the receptors. And NSF is dynamically regulated. Inhibiting NSF has shown to prevent normal AMPAR receptor reinsertion and blocks LTP maintenance, interestingly enough. And AP2, or clathrin, it's the coat protein for endocytosis that recognizes signals like a dephosphorylated TYR876 or certain serine phosphorylation patterns on the AMPA receptor tails. Now, ARC, or ARC, is an activity-regulated gene product, which is actually not shown on the illustration above, that can enhance AMPA receptor endocytosis by linking AMPA receptors to endocytic machinery. Now, ARC, or ARC, is upregulated during prolonged activity and plays a role in homeostatic plasticity and possibly tinnitus-related changes. And together, these molecules act as a finely tuned machine, adding or subtracting AMPA receptors from the synapse to encode long-term changes. Now, if we think a little bit about ARC, which is the activity-regulated gene product that enhances the AMPA receptor endocytosis, it's very important to keep in mind that this is the exact reason, or one of the reasons, why prolonged auditory deprivation for more severe patients can potentially lead to natural long-term depression. And because it is upregulated during prolonged activity, it is pretty important, or this is one of the reasons why it's important to have consistent long enough stimulation during Susan Shore device usage, for example. This was kind of an advanced video for the knowing ones, but I thought that for those who know enough about this, it could be an interesting lesson into how the dynamics or intracellular dynamics or molecular dynamics of phosphorylation or dephosphorylation can occur, and how important the balance or tug-of-war between the CaMKII or phosphatase or calcineurin activation affects protein dynamics and is very important for LTD induction.